In this series of videos, I'm going to look at a proposed new research method that can be used in areas such as policy development, engineering, social science and healthcare. The method originated in surgery as a response to the barriers to research that exist in that discipline. The method is called null-seeking trials and is based on what's called a general randomized model. Conventional randomized trials are a widely used research method in healthcare. Null-seeking trials differ from conventional trials at a fundamental level, both in purpose and conduct, but they are related in that conventional randomized trials are a special case of null-seeking trials, and the mathematical models that they use are special cases of the general randomized model. Because the divergence between null-seeking trials and conventional trials is a fundamental one, I will start by describing the fundamental principles of randomization. If this is familiar territory to you, you may wish to skip the rest of this video. Both conventional randomized trials and null-seeking trials are based on mathematical models of reality. The purpose of mathematical modelling is to apply the flexibility and reliability of mathematical reasoning to questions that are not immediately mathematical. Familiar examples such as Newtonian mechanics take experimental data and describe them with mathematical equations that are models of the real world. The real world is observed in experiments, and the models allow us to interpolate and extrapolate beyond the experimental results themselves to predict behaviour in situations that have not been directly observed. The reliability of the prediction of mathematical models ranges from perfect so far for quantum mechanics to very good for other models of theoretical physics. Those intended to model processes that are highly complex and incompletely understood are at the other end of the spectrum, where their predictions are often doubtful. These are the models that are used for situations such as disease processes, social behaviour, education and electoral polling. Processes with an element of unpredictability are modelled using random numbers and functions of them. The study of such models is the realm of statistics. In the early years of the 20th century, statistics were successfully used to study processes like agricultural production, fermentation and manufacturing. A typical problem might be whether a certain fertilizer, A, increased the cabbage yield per acre. Researchers accounted for unpredictable variation in the weights of individual cabbages by taking a large number of cabbages treated with fertilizer A, called an experimental group, and a large number that were not treated, called a control group, and comparing their average weights. They developed simple methods to guide judgments as to whether the differences observed in the average weights of cabbages was due to the effect of fertilizer A or simply to the unpredictability of individual cabbage weights. These basic methods were applied very successfully and are still widely used today, but as they were extended into more and more areas, they began to fail. With these basic methods of comparison, if the numbers of cabbages are large, then even quite small differences in the average weights are highly unlikely to arise by chance. This makes the models sensitive to differences between the so-called populations of cabbages. But there is a problem. The model's main weakness is that there is no way of telling what the differences are caused by. If we compare the cabbage yield of two fields, one of which has been treated with fertilizer A, we may find a difference in the cabbage weights, but this may be because of a different amount of sunlight or irrigation or other factors that differ between the two fields, rather than because of the fertilizer. Getting around this problem and trying to arrive at a reliable causal inference of the effect of some specific factor or treatment remains a serious problem. Understanding how randomized trials solve that problem in healthcare gives some valuable insights into areas where randomization is not possible, but null-seeking trials may be. One way around the cabbage and field issue is to divide the fields into several smaller areas, called blocks. Add fertilizer A to alternate blocks interspersed between blocks without the treatment or with another fertilizer. The fertilized cabbages would then be mixed within each field so that any differences with other factors would largely cancel out, hopefully. Actually, this method works pretty well and is still used in agricultural research. Transferring that method to other areas is not, however, so successful. The problem of isolating the effect of a treatment from other causes has been greatest in healthcare. Early attempts to apply statistical techniques developed in agriculture and production to medical problems failed because of problems that were not at the time expected. To compare two treatments for a particular disease, an obvious approach is to take one group of patients receiving one treatment and compare them to another group receiving another treatment. For any new treatment, for example, you can note the behaviour of the disease before and after the treatment is introduced. This is the still widely used method of historical controls and is fraught with difficulty because the nature of many diseases changes over time, as does the accuracy of diagnosis, the susceptibility of populations to the disease and other treatments available. It's impossible to tell whether a difference in the behaviour of the disease at different times is due to a particular treatment effect or to one or more other factors. The historical controls method is a special case of a general principle, whereby any systematic association between treatment allocation and other influences on outcome makes it impossible to distinguish between them. Such associations cause what's called allocation bias. Allocation bias occurs when there is a systematic difference between the patients receiving the two treatments. For example, if one group is older on average, or has more severe disease, or contains a higher proportion of females, etc. An improvement on the historical controls method aimed at avoiding allocation bias is to take two alternative treatments, standard and new for example, and a series of patients with a specific disease. The first patient is given the standard treatment, the second receives the new treatment, the third the standard, and so on. This is a promising method, but when put into practice it failed. It turned out that doctors often had existing beliefs about which treatment would be more effective. A patient who was improving spontaneously or responding to standard treatment would be more likely to be enrolled in the trial if the next treatment was standard than if it was new. Similarly, a patient who was deteriorating on standard treatment would be more likely to be enrolled if the next treatment was new. On average, this means that patients receiving standard treatment tended to be improving, but those receiving new treatment tended to be deteriorating, so had more serious disease. This type of behaviour, where doctors and patients go to considerable lengths that have the unintended effect of thwarting a research project, seems perverse. Nevertheless, it is a major problem in medical research and was only overcome by imposing extreme measures. To see why this was such a problem, we need to consider research from the point of view of the doctor and patient. A patient with an illness is concerned for their health. They have limited knowledge and experience of their condition and must put their faith in the judgment of doctors. By their calling and training, doctors put the patient's interests above all else, it is hoped. Though they may be participating in a research project, that is of secondary importance. If they perceive an advantage to the patient from giving a certain treatment, then they are bound to advise the patient accordingly. It would be unethical and professionally improper to withhold any such impressions. Doctors are well educated in the field and are likely to have wide experience of the behaviour of the illness and its treatment. If they're participating in a research project on a particular treatment, they cannot help noticing whether patients on the new treatment appear to do worse or better than other patients. If they do notice such a difference, then conduct of the research project is a lower priority than securing what they perceive as the best treatment for their patients. This then leads to a conflict of interest between the aims of a research project and the doctor's clinical judgment, and usually clinical judgment wins. From the point of view of research, doctors have proved so resourceful at creating allocation bias that the only way around it has been to make it impossible for patients or doctors to interfere with treatment allocation. This is achieved by using a system of allocation that they can neither control nor predict. The necessary and sufficient condition for allocation to be unpredictable is that it is made at random. Randomization not only defeats doctor-mediated allocation bias, but it also prevents all other forms of allocation bias, whether they are known about or not. This necessary and sufficient condition means that if you want to avoid allocation bias, you have to randomize. There is no other way. This fact has made randomization a standard in medical research, but in practice it's not always feasible. And when it's not, evidence is unreliable because of potential allocation bias. In fact, one purpose of null-seeking trials is to make randomization feasible in areas where currently it is not. In truth, there is one qualifier about allocation bias. The nature of random allocation means that it is still possible to be unlucky with randomization, leading to the phenomenon of accidental allocation bias. This occurs if the randomization process happens to allocate more poor prognosis cases to one treatment than the other. There are widely used methods for minimizing the chances of accidental bias. They work by arranging for equal numbers of particular types of patient to be randomized to each treatment. Such methods are effective at reducing accidental bias that concerns prognostic factors that are known about, but they have no effect on the chance of accidental bias that concerns factors that are unknown. The only way of reducing accidental bias from such unknown factors is to increase the sample size. Anyway, from the point of view of the patient and doctor, making a decision about treatment at random, without the input of the patient's preference or professional judgment, is a significant imposition. The primacy of the patient's best interest over the interest of the research project is a fundamental principle, and the issue of when to allow randomization is one that requires careful independent scrutiny, which is why all clinical research needs the approval of ethical committees. In fact, there is another kind of bias that randomization does not address. This occurs if there is any association between the treatment allocation and the outcome of the condition other than via the direct effect of the treatment. This arises after allocation and is called post-allocation bias. An important example is the placebo effect. Inert tablets are known to have a beneficial effect on health by the power of suggestion. A trial comparing any tablet with no treatment at all is likely to find the tablet effective even if there's nothing active in it. We're not interested in this effect as much as the chemical action of particular medicines, so the placebo effect confounds our result. This kind of bias is not avoided by randomization. In fact, for post-allocation bias, there is no equivalent to a randomization that avoids bias of known and unknown causes. It's addressed in different ways depending on the specific situation, but it does remain a common problem and a reason to doubt the findings of a trial. In the case of the placebo effect, the normal solution is to ensure that neither the doctor nor the patient knows which treatment they're receiving. Methods of doing this are known as blinding. Single blinding means the experimental subjects or patients don't know which treatment is being received, and double blinding means that neither the experimental subjects or patients nor the people who measure the results of the treatment know what the allocation was. In practice, this is usually done in drug trials where 2 batches of tablets are manufactured that look and hopefully taste identical, but only one of them contains the active drug. The placebo effect then acts equally in both trial groups and does not bias the difference between them. So that's the situation with randomization. It's the only method that effectively deals with allocation bias, but for ethical reasons it is often not possible, and it does not deal with post-allocation bias. I'll look at conventional randomized controlled trials in more detail in the next video.